Hello, everyone. Welcome to today's live broadcast, Flow Cytometry Data Analysis in a Flash. I'm Christina Jewell of LabRoots, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots and sponsored by Beckman Coulter Life Sciences. Beckman Coulter is dedicated to advancing the optimizing of the laboratory. The company's global leadership and world-class service and support delivers sophisticated instrument systems, reagents, and services to life science researchers in academic and commercial laboratories, enabling new discoveries in biology-based research and development. For more information, please visit www.beckman.com. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window, or you can submit your problem through the green Q&A button on the lower left. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located at the bottom left-hand corner of your web page and follow the process of obtaining your credits. I would now like to introduce today's speaker, Dr. Joyce Slusser. Dr. Joyce Slusser is a senior scientist at KCAS Bioanalytical and Biomarker Services. Dr. Slusser recently joined KCAS in 2016, bringing with her the knowledge and expertise to expand KCAS's flow cytometry and tissue culture offerings. Her proficiency in flow cytometry stems from years in the lab at 2 medical colleges and as a consulting scientist in a corporate setting. Over the past 20-plus years, she has gained experience across multiple species and platforms, both in research settings and in clinical settings. Her education is rooted in immunology, virology, hematology, and oncology. Joyce has published over 15 articles and is an active member in the International Society of Analytical Cytometry, International Clinical Cytometry Society, American Association of Immunologists, and founder of Great Plains Analytical Cytometry Association. I will now turn it over to Dr. Slusser for her presentation. Dr. Slusser?
Thank you so much for the introduction, and thank you to LabRoots and Beckman Coulter for facilitating, and welcome all. I would like to jump right into the presentation because I do have an extensive software demonstration I would like to do. So let's get rolling. My first slide is an introduction to the why KCAS and how we came to this presentation today. So first, KCAS has been in business for about 37 years doing pharmacodynamic and pharmacokinetic studies. It's a contract research organization, meaning we run these experiments or studies for our clients who are by and large pharmaceutical companies. And biosimilars and biomarkers have recently jumped to the forefront of pharmacological product development. So things like antibodies are now being used as drugs, which means we are doing more studies on biomarkers now than just the drug and its metabolites. So as a contract research organization, we operate in a GLP environment, which is regulated by the FDA. So GLP means good laboratory practices. These are very well defined, and there are several publications, including the Crystal City Guidelines, which cover how a study should be conducted and documented. However, because biomarker studies are so new and flow cytometry even newer to the scene, they are not covered specifically in the Crystal City Guidelines. So we apply as many of those guidelines to our biomarker studies as possible. We receive study samples from across the globe, and one of the things that is critical is the stability of those biomarkers during transit, over holidays. There are always delays in shipping. So one of the first steps I undertook was to characterize some of those biomarkers we would be offering in our portfolio during transit and stability studies. Right? So I, for flow cytometry, will predominantly offer services on whole blood, but also occasionally bone marrow and CSF and things like that. So peripheral blood will be collected in a number of ways, but most commonly those are potassium EDTA, sodium heparin, ACD, and a new player to the market is the BCT strep tube. If you're not familiar with that, it is an EDTA anticoagulant with a fix in the tube. All right, so that allows the cell to be stabilized, but it also introduces a new point of contingency, which is that my antibody must recognize not only fresh epitopes or fresh markers on the cell surface, but fixed markers in order for the side-by-side comparison of a fresh epitope to a fixed epitope. And those can change over time. So we set a baseline for flow cytometry of these biomarkers at 4 hours, and then we sampled these again at 24 hours, 48 hours, 96 hours, 144 hours, 192 hours, and 240 hours. That will offer up to 10-day stability on these biomarkers. All right, no one panel will fit everyone's needs. So we have 5 different panels that we initiated. These are the Duraclone immune monitoring panels available from Beckman Coulter. The first would be TBNK. It offers a plethora of small subsetting, right? So CD45 gating to CD3, you'll see 4s and 8s. You can pick out NK cells with the 56 and the 16, the B cells with the CD19. So you can overall get a picture, a snapshot of peripheral blood cells. You will notice that it's a 10-color instrument. I have 2 open channels listed. That allows my client to choose 2 markers that will specifically address something that they are monitoring. Then I have a full 10-color panel for T subsets, so using CD45RA, CCR7, CD28, PD1, etc., to characterize further T-cell subsets. Or maybe you just need a panel that looks at T-cell receptors. We also have a B-cell tube offering IgD and M, so you can know about lineage specificity of the B cells, and then a dendritic cell. So again, just to cover the bases, we started with this basic set of markers. So 5 tubes offering a minimum of 8 markers per tube, 4 collection media, and 9 time points. So for each donor, this gating on just % positives versus negatives, that's 6,480 data points per donor, and we decided to look at 3 donors in order to have a full set of information so I could do some statistics. All right, so the goals of my presentation today are to tell you how to manipulate that 6,500 data points per donor, link gates across study files so that each file is analyzed in the same fashion, generate reports, and do this in a GLP fashion that allows you to retain original data and further subsetted data or reportable data or statistics, however you wish to term that, to increase the throughput of those 7,000 data points and maximize output using something called composites. Right, so without any further ado, if you'll give me just a moment, I am going to start sharing my desktop so we can jump into the software. All right, so hopefully you will see a screen in front of you that has dot plots and histograms. And as I am no longer looking at the window that gives me feedback, I'm going to assume this is the case. All right, so what I've loaded on the left side are a number of tubes, and as I hover over them, you will see that some of them have thousands of events, and some of them have up to 1 million events. So some of these files are quite large. We're going to start at the top of the list, but just for demonstration purposes, if you were ready to load your data set, you would click on the little red Kaluza ball and open files and import them. So my first set of data are going to be quality control samples for my flow cytometer. And the first of those is the flow set bead. This is a bead of defined fluorescence for me, and I have selected channels in my dot plots that I target my beads to, and these targets allow me to optimize my voltages on my PMTs in order to see negative populations and positive populations fully contained on my 4-log scale down here, and always have my data in the same place. So if I want to manipulate hundreds of files and have them all gated similarly, I need the populations not to bump around the plot. Additionally, if I want to draw any conclusions about regulation, upregulation, or downregulation of a marker, I need those populations to remain fixed with respect to each other. And I'll show you what that means shortly and how I document that. All right, so suffice it to say, I look at single beads. I have a defined area or target where those beads must appear. The instrument actually automatically adjusts the voltages to get those beads into these channels. All right, then I run my single color control tubes. All right, so there are 10 of those plus an autofluorescence tube. So just really quickly, I'm going to grab a couple of files and show you what that looks like. Right, so as the software opens that, it will present a window that says, what do you want to do with all of these tubes? I can click on a single tube and analyze it individually, or I can do some other actions. So today's action is add selected to a new compensation. All right, and if I would choose that, the instrument will, or the software will look at the data and say, okay, you have this many tubes. I need you to tell me, is this FL1, FL2, FL3, FL4? So if I would click this, some of these are not single color controls. Where it's able, it will assign a channel. These two it's not going to be able to assign for sure. But it finds this one in the correct channel and this one in the correct channel, so that's all good. Then I have 3 choices here for the method of doing compensation. The first is positives only. If you use CD45 or CD4 and you use it in every flavor, then you'll only have positive cells and no negative cells to gate on. So CD45 would be a positives only. If you have a population of cells that have high autofluorescence, you may want to do an autofluorescence subtraction, and you have that choice. For me, none of my markers are expressed globally, so I will always have a positive and a negative population in my dot plot. So I always choose the positive, negative, and then you click Generate Compensation. All right, so if you think about what compensation looks like for a 4-color instrument, this is where I started. It's the FITC channel versus the PE channel, the FITC channel versus the ECD channel, and the FITC channel versus the PC-Sci5 channel, for instance. That's not too difficult of a compensation to set up. When you have 10 colors in a tube, this is a 10-color panel, I now have, instead of those few plots, a 10-color panel has 56 dot plots to evaluate for compensation. That's a lot of tweaking. So I always start with my auto compensation using the software to get global settings that I can apply and tweak if necessary. All right, so this, regardless of what order they're listed, across the bottom of my screen you will see that they're listed in the order of the FL channels. So I'm going to close this tab to the left so we have more real estate here. All right, FITC is along the bottom of each of these plots, so everything is plotted against the FITC channel. And the next thing I'm going to show you is by clicking on one of these boxes, I can globally adjust this gate across all of the FITC dot plots. So here are where my negatives are on all of those dot plots. And I can grab this one and adjust, these are all linked gates, the gate around the positives. Right, so once I adjust my gates, I have negatives and positives. Now, the software is going to look at the mean of this population, actually the median of this population on the Y versus the median of this population on the Y. And so you can see those are very dissimilar and compensation will be required. I have one weird little box down here. So if you'll remember my beads, not every channel has a target right in the middle of the PMT. Some fluorochromes are extremely bright. Some fluorochromes are extremely dim. So if I use the same settings for a really bright fluorochrome as a really dim fluorochrome, for the dim fluorochrome, my negatives will be below 0, and my positives will be somewhere in the middle. For the really bright fluorochrome, my negatives will be too high, and my positives may be off scale. So each of these is optimized for the fluorochrome being used, and some of the targets are quite low. So let's go back to this compensation. So that's reflected here. While the 50 is the same across the bottom of the plot, you'll notice on the Y that the populations are not in the same place. So these are kind of high because chrome orange is kind of dim. These are kind of high because PAC blue is kind of dim. They're both excited by the 405. These are smashed up against the axis. I've expanded this one in something called logical. So if I get here, I have what BD users would call bi-exponential and other users would call logical. I can plot below 0 what those populations would look like. These gates are no longer linked, but if I return them to where they should be with logarithmic, they are now linked. All right, so for really bright fluorochromes, negative can be really negative. For really dim fluorochromes, negative can be fairly bright. It's just part of using all of these different fluorochromes together and making them play nicely. So I will have a tab then for all of the fluorochromes that I'm utilizing. So in this case, FL2, PE is across all of my x-axes against my y's. And a compensation matrix will be generated based on the calculations required to bring these medians for the populations together. Okay, so running a 10-color experiment and all of those dot plots, 30, 50, 56 dot plots, that's a lot of data. And if you had a subpopulation that you wanted to draw a box around and say this is my stop, every time the instrument runs a sample, at the end of that sample, it will spend time putting all of those dots where they're supposed to be and updating all of those statistics at the end of the run. And you can't go on to the next sample until that's done. So the data processing bit during acquisition can really slow down your acquisition. That's not my goal. I want to acquire the samples as quickly as I can once they are stained and ready to go, and then do my analysis offline. So using my flow set beads to set my targets, using my single color controls to generate my compensation matrix, I can jump right into my first sample as soon as those are accomplished. And I don't need very many beads, nor do I need very many of these single color controls. So my first sample that I run, I'll have a few gates and mostly histograms and maybe an informational plot just so I can keep track. All right. Not a lot of boxes to populate before I move on to the next sample, but all the information I need to see in order to know if the sample was prepared properly, lysed properly, and is running correctly. All of that data is collected uncompensated because that's another process that the instrument must do while it's running and acquiring that I don't need it to be doing. So my data is collected ungated and uncompensated. So I've generated a compensation matrix here. I'll go to the Kaluza ball and I'll say save selected as and choose compensation and give it a name. When I arrive at my first data set, I can apply that compensation or I can apply it across every data bit in the set. Right? And these histograms are not very informative. So I am going to create the template of reporting that I want with all of the correct gates, and I'm going to add that. And so I do that by just adding a new sheet. And on this new sheet, I can save anything that was useful from the first page. So I can copy. And now I have two choices for pasting, paste and paste as a link. If I paste, this sheet is independent of the other sheet. If I paste as a link, what you'll see is this little chain link in the corner of the box. And it means that when I move something on this dot plot, it's also changed on my original. So it's a way of linking together gates across pages. All right. So I will create a report sheet that gives me a down and dirty, something that maybe the client needs to know how the sample will they proceed with the next collection on that particular individual. So I create those templates, and here is one. This is an empty template, and it says drop data set here. You can't read that part. So I have two options. I can drag my data set into an empty protocol and populate it. So now I have multiple sheets and I have dots in the box. Or I can take the data, take the protocol and drag it onto a data set that I just collected. And now it's no longer a single imported tab, but my reporting tabs as well. All right. Looking at this reporting tab is my first opportunity to look at my compensation. So this is an uncompensated. How do I know that? One, the compensation matrix is all 0. 2, my CD4s should not be up in the box where my CD8s are. These should be independent populations. They should not be co-staining in a single individual, a human. So now I'm going to import the compensation that I just made with my compensation matrix. And these cells will now go into the box where they're supposed to be, and my compensation matrix is populated. So I use my single color control to generate a compensation matrix, which I applied to my sample. I built a protocol for reporting all the data bits that I want to. It has, who ran it, where it was run, and some statistics. And I have something that I can quickly export to a PDF, print selected, and I'll say my current sheet, this report, as a PDF, and I can drop that into a box for my client to look at, comment on, can you move a gate, and we can quickly update any statistics. If they have questions, I can add boxes pretty easily. Now, I have multiple data sets over multiple times on multiple tubes. How do you handle that? Okay, so I'm going to scroll down just a little bit. All of these data sets are just the EDTA collection over a period of time. My 4-hour, my 24-hour, my 48-hour, 96-hour, 116 hours, 192 hours, 240 hours. That's just the EDTA on an individual number 1. How do I put it together and make any conclusions? Well, I could and make a report. Or I create a new sheet and have my phenotyping tab be a report of all of the statistics associated with my history. Right? Because over time you don't expect that the populations will stain perfectly. So I've just created a few examples here, and I will show you how I did that. So on this toolbar here is something called an information plot. You click on that and you get a little table that allows you to scroll through your FCS header and pull out bits of information, the title of the study, when it was run, who ran it, etc., the time, date. Then I can go to my data set and choose any gate and any statistic collected. So the number of events, the percent in that gate, the total, mean, CV, standard deviation, that, all right? So I have a gate. It has some events in it. I know the fluorescence on those events. So this is the median of the CD14 negative in the FL5 channel, which is CD14. This is the median for the CD14 positive in the same channel. So if I want to know how intense the signal is over the background, the negative population, I create, this is an Excel spreadsheet, I create an equation. And that equation is the median of the CD14s on the FL5 parameter divided by the median of the negative in the same parameter. So for data review, someone with a calculator can say 5.06 divided by 0.25 is 19.93 and know that the calculation is correct. But I don't have to do it. And when I run the next tube, I can apply the same data sheet to that file. So CD4 signal to noise is 19.93 at 4 hours in EDTA. At 24 hours, the signal to noise is 11.87 for the same signal. Can you see that visually? Yes. Is the percentage changed? Not significantly, from 4 to 3. But there is a dimming of the marker on the circuit. At 48 hours, that signal is 1509. Still not substantially reduced in number, but starting to go away. Now I've nearly lost the CD14 positive population, even though those that are still there have a distinguishable signal. So I have now doubled the data set from 6,480 data points to 12,960 data points, because not only am I reporting the percent positive, but I'm also reporting the signal-to-noise ratio for the markers, for each of the markers in that tube. So you can start to see that the data analysis and the data points generated gets a little bit out of hand when you do this for a number of samples. And all of this data has to be exported to Excel for comparison, right? No. And I will show you how. So this is that data set. You'll see that this is the 4-hour, this is the 24-hour, this is the 48-hour, and the 96, etc. So the 9 time points in the EDTA for the first donor for the TBNK panel. And I've done something called composites. So instead of being able to click on each file individually and view it, what I have is a list of files and some boxes I've selected. And again, just like the single color controls, all of the tubes are listed across the bottom of the page because I made report sheets. So I can go visit them individually and look at the information just as it appeared on my individual file. But then I can click over to my composite and look at each data set, which is linked to the original, and look at the gates and the data together. So here's the 4-hour. Let me close this. Here's the 4-hour data set. Look how pretty those monocytes are out there at 4 hours. They still look pretty at 24 hours. They still look pretty. There are fewer of them, and they're starting to trail off by 48 hours. Virtually 50% reduction in signal and 50% reduction in number positive. Losing them, gone. So EDTA is not the happiest place for monocytes to hang out after a couple of days. So you wouldn't want to do a monocyte study in EDTA collection because within 4 days all of your signal is gone. All right, now I have all of these data sets together. I have an Excel spreadsheet associated with this data set. I can also now do things like compare data and gates across the data set. So I click on the comparison plot and it brings me this box. If I make it a little bigger, it'll get its own space on the plot sheet. So here's my data set, 9 data points. I want to measure the mean of the population, the deviation of the population, the percent gated, etc. So let's just choose the
Hi, everyone. It appears we are experiencing a little bit of
technical. It seems I was disconnected. I hope you can hear me now. I'm going to jump back to the screen. Just to make sure. We lost audio, and it looks like we're back. All right, so what I did was choose the number of events to plot against various gates. So this is the B cells, the CD19 positives across the subset. I can add the CD4, CD25s across the subset. I can do this as a graph, but I can also do this as, oh, got thrown off my game there, as a bar chart, right? I like my bar charts rather than graphs, but it's up to each individual user, all right? So what I did in my first table is group those TBNKs and how long they persist in EDTA. In my second, the monocytes. In my third, I'm looking at class 2 on each of my subpopulations and how long it persists. What percent of the population stays positive? Right, this is the one we just created. Right, so let me jump back to the analysis list. And the first set that we're looking at is the EDTA. So now this is the heparin. What do those subpopulations look like? Well, let's have a look. I find this to be the fastest way. I can create the template once and then drag and drop data into my template. So this one is 360 hours 1-4. If you read through that label, I can drag it into this box. Now if I scroll back up, you'll see that one of these things is not like the others. This is a 360-hour time point of 1-4, where this is a 24-hour time point of 1-1. This is donor 1 EDTA. This is donor 1 DCT2. Right? So once the template is built, I can drag and drop data. This is going to speed my analysis on the back end for reporting. So in a GLP environment, you do a first phase, method development, where you tweak all of your reagents and you tweak all of your settings and you tweak all of your templates. And once everything is where you need it to be, we lock it down. We proceed to phase 2, which is sample testing. Right, so now I have a template. I do my sample testing. I drag and drop my data into my templates, and I'm on a roll. So I kind of sidetracked myself there by showing you how to repopulate that, but that's exactly what I did. I built this one. And moving on to the next slide. All right, so real quickly to recap, we covered generating a template, labeling of data files, zooming. We did not cover zooming into data. We did cover resolution of data. We can go back and cover zooming. Linking data sets, i.e. creating a composite. Linking gates across the data sets in those composites. Using the information tool to create the statistics at the bottom and generating an Excel spreadsheet. Creating data tables and graphs using Kaluza. Creating reports, and then exporting data to Excel. So I see that we missed covering exporting data to Excel. So I'm going to go through the software. All right, so I have this really pretty table, and I have all of this data. Can I export it to Excel? And the answer is yes. Actually, I'm going to go up to the top of the page. So I have this summary table with calculations. This is known as a hierarchical gate, so I have the relationship between these populations described in Boolean gating here. I'm going to click on the red ball, and I'm going to say export. And now I can do selected statistics, the statistics from this current sheet, any report sheet, all of the sheets, or multiple sheets. So this will take me to a CSV sheet, right? So CSV outputs, and I can save the data from this file. So that's exporting statistics. The other thing I didn't cover was the zoom tool. So let me find, here's a dot plot, and we want to know more about these cells at the bottom. So I'm going to click on the zoom. Relatively big file. And you're able to, doesn't like all my windows open, to zoom in on populations of cells. There, now it's going to do it. Right, so it's changed how my picture is displayed, and I've zoomed in on a subpopulation of cells. Right, so there's a zoom in and a zoom out tool. Sorry, I double clicked. So that you can go in and look at subpopulations of interest. That's important if you're doing things like stem cells, and they're 0.01% of the subpopulation. Right? It will maybe help you to draw a better gate if you can zoom in and click around the subpopulation that you want. Okay. We'll go back to the presentation. Right, so creating reports, zooming, linking, exporting to Excel. Right. So now I've made some tables for you. I have an extremely large data set, and some of these files have over 1 million events. Getting through all of them for all 3 donors was going to be extremely difficult. So we just zoomed in on the TBNK, which is quite useful to most people. And then we created this summary slide for this presentation. So the donor number 1 collected an EDTA across these 9 time points. We were able to draw some conclusions by subpopulation percentages. So overall integrity was about 48 hours. I start to lose my entire lymph distribution, so percent positive deviates substantially after 144 hours. So we can look at individual markers or subpopulations and draw conclusions. So EDTA, you need to get it into the laboratory and look within 48 hours to 96 hours if you really want to know anything about the population. If you use heparin, probably won't surprise you that this sample is stable for a little bit longer than in EDTA. Right? So macrophages and monocytes especially don't like EDTA, but they're a little bit happier in heparin, and the populations are stabilized for a little bit longer. Some of the samples or some of the markers don't look good after 26, but overall you can go from maybe 4 or 5 days. If you collect in the BCT tube, that's the tube with EDTA and FIX, most of those markers last well into the weekend, right? And the distribution is pretty clear out to 360 hours. I didn't display all of that data past 240, but the tube hangs in there, except for the CD25, pretty long, right? So collecting with a FIX in the tube, if you're just doing T-cell monitoring or B-cell monitoring, you are good. Right? If you collect in ACD, then you're also going to last for a substantial amount of time, right? Most of the distribution anyway looks good, clear out to 360 hours. So if I was going to do something where I needed to do stimulation, etc., in order to collect cytokines or information about signaling or treatment in a Petri dish in the laboratory, I would want an ACD tube. If I'm going to do a study and just report out surface or intracellular markers, I'm probably going to go with a BCT tube because it's fixed, and I actually received samples that were frozen accidentally, and because they had been fixed, the epitopes were still pretty good and the sample looked pretty good. So I'm going to report on in my GLP fashion that the samples were not stored appropriately during storage and shipment, but that the markers did look pretty good when I received the samples. All right, so my conclusions. I'm going to wrap this up. The toolbars in Kaluza, as you saw, I could do things like get the wheel with all of the tools. That's a simple right-click on any plot or box. They're easy to access. They're pretty intuitive. I was pretty impressed with the software development team. They sit you in front of a computer. They open up the software, and then they give you little tasks to do. And they put a little camera on the laptop, and they watch how you search the screen for things and where you expect them to be. And the next time you go in to beta test the software, those tools are right where you went to look in the first place. So I have to say it was the funnest software development experience that I've ever had. And I was really impressed each time I went back to try the software that the tweaks had been made. So it's quite intuitive and straightforward to use and makes me happy. I love flow cytometry, and this software makes me very happy. We covered creating templates and using them in a reproducible fashion for analysis. We talked about manipulating large data files and data sets. So regardless of how many statistics I pile onto this thing, the templates open up, they repopulate, I move a gate, it updates my statistics. I have a profound ability to crash software and break things, and it's tough for me with Kaluza to find a way to break it. I've done it once or twice, but they've troubleshot right through it. All right, so we looked at merging files, making composites, linking data sets, linking gates in order to standardize your workflow and your data reporting. And then we talked about incorporation of tables into the report format to allow for quick reprocessing, but also straightforward data review. There's no look at Excel and then look at the plot sheet to see that the numbers match. Look at the Excel and then look at the plot sheet to make sure the numbers match. And then look at the calculation because it's all on the page. And in 3 pages of PDF, you can straightforward review all of these markers and statistics. All right, so I really would like to thank you for participating today. It's been an awesome experience for me to give this presentation, and I will let Christy, my moderator, read some questions.
Thank you, Dr. Slusser, for that informative presentation. Now, before we do get started on the Q&A session, I would like to remind our audience how to submit those questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We will try to answer as many questions as we can. Okay, Dr. Slusser, our first question is, does your machine do auto voltage based on the beads, or do you make the adjustments manually?
I can do either. It is a Beckman-Coulter Navios 10-color instrument with 3 lasers. So I can use the beads to acquire settings, or if I'm just setting something up for the first time with development, I use my antibodies to tell the story. And then I tell the beads where I want them to be, and then I use the beads to standardize the data set. So it can go either way.
How do you add text and graphics to the report sheets?
Oh, so I skipped that. And I won't exit because it takes time, but there is a cute little icon that's called text box, and you can open the text box, and for instance, I can import the KCAS logo and put it on all of my data sheets. You can also type in the box. I have created for other users places where they can write comments, etc., or sign and approve things. Text box.
Is it possible to see an example of autofluorescence subtraction?
I do not have any, but I am sure there is someone at Beckman-Coulter who has run something that is ugly that could give you a demo. If you're in an institution with a flow cytometry core, they can probably get you samples. And I should mention, Kaluza will open just about anything that you collected. So it doesn't matter what instrument you collected it on. If it's in the LMD FCS format, it can be opened and analyzed.
Are the numbers in percentages or MFI?
However you choose. So you can do median, you can do arithmetic mean, you can do geometric mean. That was one of my jokes I was going to tell at the beginning of the presentation, right? I'm in Shawnee, Kansas, which is in the arithmetic and geometric mean of the North America, if you consider how many people are on each coast. It's not a very good flow cytometry joke, but there you have it.
Can you explain also if it's possible APS diagrams?
I don't know what that is.
Okay, we could probably answer this offline if we get the answer to that.
Yes.
Yeah, so our audience member can submit a more specific question, be more elaborate on that.
Yes.
Dr. Slusser, what would the stability of CD34 cells be in the different types of additives in the collection tubes?
I think CD34 is pretty stable and would be BCT fixable. Again, it's more about the antibody that you choose, recognizing a fixed epitope. So we were pretty careful in our selection of antibodies for recognizing the fresh and fixed epitopes so that we could get the same answer across data sets. But I'd be more than happy to give it a try if someone wants to send me some information.
How would you get the percentage of Boolean gate?
The same way you get percentage of any other gate. So it will do a percentage of parent, right? It's what's going to be reported on the statistics bar. So if it's leukocytes and lymphocytes and CD3 and CD4, it will be of the parent.
Is there any specific voltage value for each cell line, or does it depend just on the analysis protocol of search cell markers?
So settings will be different for doing apoptosis than from doing surface markers than from doing intracellular because you've done different things to the cell. So I would say no one set of settings fits everything, but you can create some generalized settings that work for most things.
Okay, Dr. Slusser, we have a question that came in. It says automatic separator populations. I believe they're asking for. Go ahead.
It says auto gating. I'm going to let Beckman Coulter take that question. There was a really handy tool that they've just created that one click and you get positives and negatives on a histogram. There are auto-gating tools in the software for acquisition. The compensation matrix comes up with an auto-gate, but I don't know that there's auto-gating in Kaluza specifically. So I will let Beckman Coulter follow up on that question because I'm not really sure.
Very good. Let's see. We have so many great questions coming in.
I saw one that says, is it single user or multiple user? And the answer is yes. There are both. So you can get a single license for a single individual. You can get a network license for your institution. The institution comes with a number of seats. First in, first, occupies the seat. So you log in, you log out, and you always have X number of seats available for people to use.
Hello.
So it looks like Beckman Coulter is going to reply to the APS question.
Yes. This is Laura Pajak. I am the product manager for Kaluza, and a couple comments. Kaluza at this point does not have auto gating. It is on our roadmap for future builds. To follow up with respect to the question that Joyce just talked about with Kaluza versions, there are multiple formats of Kaluza that are available. They range from a single user, one-year license, or a perpetual license up to multi-user network licenses that Joyce described. And each of these are priced at either for commercial or for academic institutions.
Very good. Thank you, Laura, for clarifying that. And I believe that we are out of time for any more questions, but we'd love to open up Dr. Slusser for any final comments that you have for our audience.
I have used a lot of software in 30 years and been through a number of revisions of softwares out there. I was a dyed-in-the-wool true fan of several other versions until I met Kaluza. And I have to say I fell in love with even the first version, which was pretty clunky. But as I said, they were very responsive to needs, wants, and desires for the software. And I fall a little bit more in love with it each time it comes out. So, and the same is true for instrumentation. I have run a lot of instrumentation over the years. And the fact that there's one software that will open many, many different versions of a file, and I can set gates on my data collected on my Accuray and turn around and save that protocol and apply it to a different instrument, like just put that protocol onto data from a different instrument and have the gates in the same place. Makes me very happy. It's extremely flexible. So that would be my parting shot.
Thank you again, Dr. Joyce Slusser. I would also like to thank our sponsor, Beckman Coulter, for making today's educational webcast possible. Today's webcast will be available for on-demand viewing through June 9th, 2017. You will receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. Thank you for joining us, and we hope to see you next time. Goodbye.